All right. So today we're going to continue with psychology and anatomy, and in this class we'll do the central nervous system, and as well as we'll do central nervous system as well as muscle contraction. So let's start with the anatomy and psychology of neurons. So yeah, let's start with the anatomy of a neuron. So cell body is this entire thing right here, is has the main nucleus and cytoplasm, and also contains mitochondria, ribosomes, and rough endoplasmic reticulum, Golgi body. So this cell body has the nucleus that you see right here. It also has a ribosomes and mitochondria, Golgi body, all of that stuff. So the cytoplasmic process are thin cytoplasmic extensions of the cell body. And these are the dendrites. The dendrites that you see right here are the organelles that carry impulses towards the cell body. So impulse will uh, come towards the cell body and then move through like this through the axon. And the axon, this entire thing right here, is the tail, or the axon is the organelle that carries impulse away from the cell body. So away from the cell body to the next synapse. Um, and they carry impulse away from the cell body, and they are enclosed in something called the myelin sheath. So this yellow stuff that you see are called the myelin sheath. And the axon terminal is this thing right here. These are the terminals that have many mitochondria, synaptic vesicles, Ca2 plus channels, calcium ion channels. We'll talk more about this. And they are in connection with the junction of different neurons. So these axon terminals are connected to different other synapses as well. So different neurons as well. So the job of axon terminal is to get connected with the synapse of another neuron. And a synapse includes a synaptic cleft. A synaptic cleft is the gap between the two neurons, and this synaptic cleft has different enzymes to break down neurotransmitters. And as well, a synapse also contains a postsynaptic membrane. A postsynaptic membrane is the synapse that is present after the impulse is carried. So, for example, if the impulse was carried from this neuron to this neuron, then the synapse of this neuron would be the postsynaptic membrane. All right. And this postsynaptic membrane has receptor proteins for neurotransmitters. So these neurotransmitters can bind to the receptors present on the synaptic membrane, and then the impulse will be carried forward. And moving on with the new anatomy, myelin sheath is this compound that covers the axon, and it insulates the axon of many neurons and speeds up the conduction of nerve impulses. So the job of myelin sheath is that it allows axon to increase the speed of conduction so that impulses can travel much faster than normal. Right. And this myelin sheet is made of something called the Schwann cells. I can't really pronounce this, but yeah, you should memorize this. And these cells have a nucleus, remember. All right. So let's move on to the reflex arc. A reflex arc is the involuntary, fast, and automated response from our body. And the advantages of a reflex arc is that it's fast, it's automatic, it's involuntary, it doesn't involve your brain, it's instinctive. That means there will be always the same response every time, and it protects you from harm. So, for example, an example of a reflex arc is uh, is when you touch something that is hot, you'll move your finger away instantly. This is called a reflex arc. And how does a reflex arc work? First of all, you'll detect a strong stimuli by the receptor. So, in the case of touching a hot thing with your fingers, the receptor here would be the thermoreceptors. So, the thermoreceptors detect the strong stimuli, a change in heat, and an action potential is generated in the sensory neurons. So, this sensory neuron has a synapse with a relay neuron, and this action potential is then carried to the relay neuron. Action potential is passed to the relay neuron, and this relay neuron has a synapse with a motor neuron. So, a synapse with a relay neuron means with a motor neuron means that action potential can be passed on from the relay neuron to the motor neuron. And remember that motor neurons motor neurons are always connected to a muscle junction. All right, so this motor neuron is has a synapse with a neuromuscular junction. So this motor neuron then passes the action potential to the neuromuscular junction, and then because of this, the muscle that is the motor neuron is connected to will have a effect. So the muscle or the effector will carry out an appropriate response. So in the case of touching a hot thing, the response would be your muscle contracting and moving your finger away from the hot thing. All right. So this is how reflex arc works. Again, this is a very basic definition. We'll talk more about this. All right. So what are impulses? Impulses are brief changes to the distribution of electrical charge across a membrane. This is also known as a membrane potential. And in our cells, the resting potential is 70 millivolts. All right. And the action potential, when an action potential is generated, then you'll have effect. The effector will be activated. When an action potential is present, then the electrical charge in your cell will go from negative 70 milliwatts to positive 30 milliwatts. All right. So yeah, action potential is when you have 30 milliwatts present in your cells. So um, what moving on with the sensory receptor cells. Receptor cells detects the stimuli. A stimuli is a change in internal uh, or external environment, and receptors are specific to one type of stimuli. For example, if you have a change in temperature, then the thermoreceptors would be activated. When you have a change in chemical potential, then the chemoreceptors would be activated. And similarly, light means light receptors would be activated. And receptors, they act as transducer, meaning they can convert any type of energy into electrical energy, because the neurons are only able to carry electrical charge. All right. So, for example, if a thermoreceptor detects a stimuli, the thermoreceptor would transfer heat energy into electrical energy, and this electrical energy will then be carried to the neurons. So chemical energy, for example, chemoreceptors detect chemical energy, and they can convert chemical energy to electrical energy. And when this happens, when electrical energy is generated, this is when you call an action potential to be generated. All right. um, yeah. So an example of receptors in your body are the taste chemoreceptor cells that are found on your tongue. So in this case, the chemical will act as a stimuli, and there are different chemoreceptors that are specific for different kinds of chemicals. So when you eat something, right, it will release chemicals. The saliva will break down chemicals, and these chemicals would be detected by the chemoreceptors. And chemoreceptors then transfer this stimuli, convert the chemical energy into electrical energy, and generate an action potential, and then do all of that stuff. All right. And yeah, so let's talk about action potentials now. Remember, the resting potential in your body is negative 70 milliwatts. And at rest, when your cells are at resting potential, there is no stimuli and no impulse found in your body. And the insides of the axon are more negatively charged than the outside. So this negative uh, milliwatts are the electrical charge inside of your cells. So if it's the inside of your cell is much more negative, then that means the outside of your cell would be positively charged. So at resting potential, always remember the inside of the cell is more negatively charged. So uh, the neuron is polarized and maintained at negative 70 milliwatts. So the neurons, the cells are at negative 70. And how is this resting potential maintained? It is maintained through a sodium potassium pump. This works in a very confusing way. So, yeah. Uh, so remember that 3 Na plus would be pumped out and 2 potassium ions would be pumped in. So 3 sodium ions move out of the cells, and 2 potassium ions move into the cell. And this is active transport. And because of this, you would need ATP. And uh, yeah. So the axon phospholipid bilayer is impermeable to potassium and sodium ions. And because of this, active transport would be needed because the membrane cannot transport any ions. So because of this, moving in and out of sodium and potassium cells, you would maintain an electrochemical gradient. An electrochemical gradient would start to build up, and an electrochemical gradient is the difference in both charge and the concentration of chemical ions across a membrane. So active transport is happening for sodium and potassium ions. Sodium ions are constantly being moved out of the cell. And because of this, the concentration of sodium ions increases outside the cell, and the concentration of potassium ions increases inside our cells. And because of this concentration buildup, diffusion happens. And so diffusion happens from a high concentration of ions to a low concentration of ions. So in this case, potassium ions have high concentration, so potassium ions diffuse from the higher concentration, meaning inside the cell, into the outside of the cell. All right, so potassium ions diffuse out of the cell via diffusion, and sodium ions, because initially sodium ions were being pumped out, now the sodium ion concentration is high outside, so sodium ions move into the cell. They diffuse into the cell via channel proteins. All right, so remember, first step is active transport of sodium ions out of the cell, and potassium ions are pumped into the cell. The next step is diffusion, because there is concentration of chemicals being built up. That means now potassium ions would be pumped into the cell. Potassium ions would diffuse out of the cell, and sodium ions will now diffuse into the cell. And all of this happens through channel proteins. So because you have more potassium ions being diffused out, there are more potassium channels open than sodium channels. All right. And because of this, the membrane becomes more permeable to potassium ions than sodium ions. This means that more potassium ions would leave the cell than sodium ions being entering. So in this case, when diffusion is happening, right? When diffusion is happening, you have more potassium channels that are open compared to the sodium ion channels. This means that potassium ions would be constantly being diffusing out of the cell. And because of this, you have more potassium ions that are leaving the cell than sodium ions that are diffusing in. And this leaking of potassium ions, because more potassium ions are now leaving the cell, this leaking of potassium ions will cause the resting potential to be maintained, which is negative 70 milliwatts. Why does this happen? Because more positively charged ions are leaving the cell. So when more positive ions are leaving the cell, because of this, the outside would become more positive and the inside becomes more negative, so the negative 70 milliwatts. And because of this, your inside becomes relatively more negative than the outside. All right. Yeah, this is how resting potential is maintained. But what happens when you have depolarization? Depolarization is when you go from negative 70 to positive 30 milliwatts. And uh, how does this happen? This happens when voltage gated potassium ions uh, closes and voltage gated sodium ions open. Because of this, there is when the channel changes shape, when membrane potential changes, and when the action potential arrives from the previous section, sodium ions would now enter the cell. So I'll explain all of this in a basic summary. So action potential arrives at the cell. Because of this, your sodium ions channel, so sodium channels open, and potassium K plus channels, they now close. Because of this, the sodium ions will now start to enter the cell, and because of this, the cell becomes positively charged, because now sodium ions are being constantly moved into the cell. So because of this positive ions being moving inside of the cell becomes more positively charged, and you get the positively charged cell. And because of this, depolarization happens. So sodium ions enter the cell, the membrane of the cell becomes less negative, and because of this, because of the change in electrical charge, your action potential is generated. So yeah, and there is another type of polarization. It's called repolarization. Repolarization is when you go from positive 30 back to negative 70. So when the action potential is no longer needed, then you want your cells to maintain that constant negative 70 charge. So because of this, repolarization happens. Repolarization is when the sodium channels closes and the potassium channels, they open again. And because of this, the potassium ions would start to move out of the cell. And because of this, when potassium ions start to move out of the cell, the outside becomes more positive, the inside becomes more negative, because more positively charged ions are leaving the inside of your cell. So because of this, you get the negative 70 milliwatt charge inside the cell. Remember that all types of polarization, detects the electrical charge inside of your body, inside of your cells. All right. And yeah, so hyperpolarization is when it's called the refractory period. And when a cell is hyperpolarized, the membrane is insensitive to any depolarization. This means that no action potential can be generated inside the cell. So why does hyperpolarization happen? Polarization happens because you want only one action potential at a time. If you have more than one action potential, this means that there can be multiple, for example, in muscle contraction, right? If you have more than one action potential at a time, action potential being generated at a time, then your muscles would be contracting a lot. And because of this, you don't want more than one action potential at a time. So because of this, hyperpolarization happens. The job of hyperpolarization is it allows only one-way transmission and one action potential at a time. Uh, yeah. So, yeah. And how is action potential transmitted along the axons? So remember, action potentials, action potential are brief charges. They are charges, they are change in charges. So delta charges. This means that going from -70 to +30, then going back to -70, all of this is called action potential. So how does action potential get transmitted? This happens through the axon. Depolarization spreads to the next region due to movement of positive ions from one region to another. From one region to another. And because of this, a local circuit is set up. A local circuit is movement of ions. Because of this movement of ions, your sodium channel opens in the next region, sodium ions enter the cell, and the cell becomes depolarized. So how does a local circuit get set up? Sodium ions open in one region, and sodium ion enters the cell becomes depolarized, and then the action potential move to the next cell, and then sodium channels open, sodium enters the cell, and depolarization spreads like this. Yeah. And how are action potentials transmitted along the myelin sheet? Remember that your axon is it's insulated with a material called the myelin sheet. The myelin sheet acts as an insulator and it insulates the axon. This stops the movement of ions where the myelin sheet is present. And because of this, the myelin sheet lengthens the local circuit. Uh, and passage of ions only happen at something called the node of Ranvier. And yeah, so remember that this is the axon, right? And so the myelin sheet would be like this. There would be small gaps between the myelin sheet, and these gaps are called the node of uh, Ranveer. I don't know how to pronounce this. Yeah, but the gaps are, remember, it's called the node of Ranveer, and the ions, they are not able to pass the myelin sheath. And because of this, the ions jump from the empty spaces. So ions would move to the empty space, they would jump from the empty spaces. So passage of ions only happen at the node of Ranveer, and action potential only happens at the node of Ranveer. Local circuit is set up between nodes. These gaps are called the nodes, and action potential jumps from node to node. And this is called saltatory conduction. So this jumping of ions are called saltatory conduction. This is when the myelin sheath insulates ions. All right. So this is how myelin insulates the axon. Uh, there is a potential, there is a brief electrical charge which is -50 milliwatts. This is called the threshold potential. And this potential is the minimum potential needed for an action potential to be generated. So if the cell, if it goes from -70 to +30, then only can action potential be generated. But if it's going from -70 to say -60, then there would be no action potential. So at -50 milliwatts, um, only this threshold potential is the minimum potential needed for the action potential to be passed on. So if the cell reaches negative 50 milliwatts, then the cell would know that an action potential is being generated. So if depolarization is greater than negative 50, then action potential is generated. So if it's greater than negative 50, meaning it's negative 30 or it goes to positive, then the action potential will get generated. Yeah. So let's talk about synapses now. Synapses are junctions between neurons as well as neuromuscular junctions. And a synapse includes the presynaptic knob. The presynaptic knob is the synapse of the neuron that the charge is coming from. And this presynaptic knob has mitochondria, synaptic vesicles that contain neurotransmitters. So these vesicles have neurotransmitters, and the presynaptic knob also contains voltage gated calcium channels. And there is also a a synaptic cleft. A synaptic cleft is this gap, as I told earlier. This gap allows enzymes. This gap has enzymes that break down neurotransmitters. And finally, there is the postsynaptic membrane. So, in a summary, a synapse has three things: the presynaptic membrane, this gap right here, and then the postsynaptic membrane. And the postsynaptic membrane has receptor proteins. Remember, only the postsynaptic membrane will have receptor proteins, and these receptor proteins are complementary to the neurotransmitters. Which neurotransmitters? The neurotransmitters that are coming from the vesicles of the presynaptic knob. Yeah, and so yeah, this is this synapse. So let me just zoom in and focus right here. So yeah, this is a synapse. So you can see this is the presynaptic knob. The presynaptic knob contains three main things: the voltage gated Ca2 plus channels, calcium ion channels, and the second thing is vesicles. These vesicles have neurotransmitters. And finally, the third main thing in a presynaptic knob is mitochondria. And yeah, why does the presynaptic membrane have mitochondria? Because remember, whenever you have vesicles, vesicle movement requires energy. So yeah, so these vesicles, right, these neurotransmitter vesicles, they diffuse from the presynaptic membrane into the gap, and from the gap, the vesicles are broken down by enzymes that are present inside the gap. And what is remaining is these neurotransmitters. Now, these neurotransmitters will then bind to the receptor protein present on the postsynaptic membrane. And this is how uh, uh, an action potential spreads from one synapse to another. Yeah. So moving on, let's talk about muscle contraction now. Remember that a muscle fiber has plasma membrane that is called sarcolemma. And sarcolemma has many infoldings, meaning it is like this. Sarcolemma has many infoldings. These infoldings are called the T tubules. And these T tubules, remember, they can conduct action potential or electric charge. So the cytoplasm in muscle fiber cells are called the sarcoplasm. And sarcoplasm has many parallel myofibrils. Myofibrils are these fibers that can be that are multinucleated, and fibers have mitochondria as well. Yeah. So this is not that important. I want to focus on this part right here. Let's talk about myofibrils. There are two types of myofibrils. Myofibrils are also known as myofilaments. This is what the IMAT syllabus expects you to know. This part you don't really need to know. So myofibrils, remember there are two types. There is the thick filament and the thick myofilament. The thick filament is made of something called the myosin, and the thin filament is made of something called the actin. And the thick filament is always attached to the M line. I'll explain what M line is, but just for now, just know thick filament is attached to the M line. The thin filament is attached to the Z line. And remember that troponin and tropomyosin are two compounds that can attach to actin. So yeah, this might be confusing for now, but it will get clear later on. So this right here is a sarcomere. What was a sarcomere? I told you earlier. A sarcomere is the muscle fiber where your muscle contraction happens. So yeah, and this right here is the sarcomere, and the sarcomere is made up of three types of lines: the M line, the Z line, and the H band. These two are lines, and there are three kinds of bands you need to know: the H band, the A band, and the I band. So, three bands and two lines. We'll talk about each in detail. So, let's start with the M line. The M line is where the myosin is attached. Myosin is attached to the M line, and the actin is attached to the Z line. So always remember M line contains this myosin, uh, and the Z line contains the actin. And so the distance between Z lines would decrease between muscle contractions. So when you have muscle contraction, when the muscle contracts, the Z line would decrease. What is the Z line? This right here, this is the Z line. These two are called the Z line. In between the Z line, you have the M line. This right here is the M line. The Z lines, remember, when in muscle contraction, they will come close together. And because of this, the distance between them would decrease. So, yeah. And the I band is known as the light band. And the I band is made up of only thin filaments. And the I band also shortens during muscle contraction. So remember, I bands as well as Z line, both of them, they decrease in distance. I bands shorten in muscle contractions. The Z lines, they decrease in muscle contractions. The H band right here is made up of light band and dark band. H band, remember, it is made up of light band and dark bands, and they only contain thick filaments. The I band was only thin filaments. The H band are made up of only thick filaments. So a good way to remember this is, remember, the letter I is thin. This means it is made up of thin filaments only. And because they are made up of thin filaments, contains the I band contains actin. And the H band, I told you H band is made up of thick filaments only. So the H band contains myosin. All right. Yeah. And remember, H is a fat or a thick alphabet. And because of that, H band is made up of thick filaments only. And the H band also shortens during muscle contraction. Yeah. So these two bands, the H band shortens during muscle contraction. Sorry, not A band. My bad. The H band and the I band. These two bands, they shorten during muscle contraction. But what doesn't shorten is the A band. The A band is made up of both the thick filament and the thin filament. So the A band contains both the myosin as well as actin. and the A band stays the same during muscle contraction. So remember, the different kinds of bands. The A band, it stays the same during muscle contraction. The H band and the I band, it shortens. So yeah, shortens during muscle contraction. The H band is made up of thick filaments. The I band is made up of thin filaments. The A band is made up of both the thick and the thin filaments. So yeah, and the Z line. Remember, the Z line has actin. The M line has myosin, and the Z line. Remember, it will also shorten during muscle contraction. So yeah, so let's start with we'll start with muscle contraction in the next class. I think the time is over. So we'll talk more about muscle contraction in the next class. But for now, you should be very good with knowing what shortens during muscle contraction and which band has which type of filaments, which line has which type of filaments. Remember, M line has myosin, Z line has actin, all of that stuff. Yeah. So yeah, I think today is enough for this class. So see you tomorrow. Bye.